Assalamualaikum and hi! So today, our group will be guiding you through a short learning session about gene expression regulation in prokaryotes and eukaryotes. We'll explain about what gene expression regulation is, what is prokaryotic and eukaryotic cells are, and how gene expression regulation work in both types. So, we hope you can follow along and understand this topic more clearly.
Alright, let's start our lesson. Hi class, let's get back what is gene expression regulation means. Okay, the first one is gene. What is the meaning of gene? Gene is a section of DNA that carries instruction to make protein. Basically, then gene is like a manual book. Okay, next is gene expression. What is gene expression? Gene expression is the process where this genetic information is used to produce protein to help the cell function. Basically, like making the enzyme or the hormone. Okay. The last one, gene expression regulation, refers to the process that controls when, where, and how much the gene needs to be turned on or turned off. Basically, this used to be like a graphic officer in the manual.
Prokaryotes are the simplest type of cells. They do not have a nucleus, which means their DNA is not protected inside any membrane. Instead, the DNA just floats freely in an area called the nucleus. They also do not have membrane-bound organelles like mitochondria or the Golgi apparatus. Because of this, their cell structure is very basic and compact. Most prokaryotes are tiny, usually between 1 to 10 micrometers. Another important point is that prokaryotes usually exist as single-cell organisms like bacteria and archaea. Even though they are small and simple, they can grow and reproduce very quickly because they don't have many internal structures to slow down their processes. Eukaryotes are more complex. Their cells do have a nucleus, and this nucleus safely stores the DNA inside a membrane. This separation allows more control over what happens to the genetic material. Eukaryotic cells also contain many membrane-bound organelles, such as mitochondria for energy, the endoplasmic reticulum for making proteins and lipids, the Golgi apparatus for packaging molecules, and vacuoles for storage. These organelles make the cell more organized and able to do many functions at once. Eukaryotic cells are also usually much larger, about 10 to 100 micrometers, and unlike prokaryotes, eukaryotes can be unicellular or multicellular. Examples include animals, plants, fungi, and protists.
Okay, so we will start on the differences on how gene expression regulation occur in prokaryotes and eukaryotes. So we will look for the first term, which is location. So for prokaryotes, both transcription and translation occurs in the cytoplasm because, as we know, prokaryotes is lack of nucleus compared to the eukaryotes. And for the eukaryotes, transcription occurs in the nucleus while the translation occurs in the cytoplasm. So for prokaryote, there is no RNA processing, but actually, prokaryote don't completely have no RNA processing, but they have very minimal RNA processing compared to eukaryotes. So for eukaryote, the pre-mRNA undergoes capping, splicing, and poly-A tail addition before undergo translation. Okay, move to the third term, which is how transcription and translation occur. For prokaryotes, the process will occur simultaneously because the operand system will occur in transcription process and the translation will process to produce protein. And differ to the eukaryotes, the transcription and translation will occur separately because there is no operand system occur. So, how we determine the gene expression regulation occur in eukaryotes? It is determined by the three steps, which is the first one is transcription, the second one is RNA processing, and the last one is, of course, translation. And the next term, we will move to the gene organization. So for prokaryotes, the genes are arranged in operands, which is the multiple genes with one promoter. Differ to the eukaryotes, genes are individually regulated. There is one gene under one promoter. And the last term, we will look at the speed of the process gene expression regulation occur. As we know, and basically, prokaryotes is faster than eukaryotes. Okay, move on to the similarities on how gene expression regulation occur in both prokaryotes and eukaryotes. So for the first one, both prokaryotes and eukaryotes follow the same central dogma. This means that the genetic information will flow from DNA to RNA to protein. So in both types of cell, DNA is first transcribed into RNA and then it's translated into proteins. Both prokaryote and eukaryote transcription produce mRNA. This transcription process occurs in three stages, which are initiation, elongation, and termination. During elongation, the RNA strand is formed in the 5' to 3' direction, and once the transcription is complete, the mRNA is ready to be used in the translation process. For another important similarity is that translation occurs at ribosomes in both prokaryotes and eukaryotes. During translation, ribosomes move along the mRNA strand in the 5' to 3' direction. For each codon in the mRNA, a tRNA molecule will bring a specific amino acid. This process will continue until a polypeptide change, which is recalled and will become a functional protein. In prokaryotes, Prokaryotes live in constantly changing environments, so they must be able to respond quickly. To save energy, they only produce proteins when they are needed. In prokaryotes, gene expression regulation mainly occurs at the transcription level. Because prokaryotic cells do not have nucleus, transcription and translation can occur at the same time. This allows prokaryotes to respond very quickly to changes in their environment. One important regulatory mechanism in prokaryotes is the operand system. An operand is a group of genes that are controlled together and work towards the same metabolic goal. Each operand consists of several parts. The first one is a promoter, where RNA polymerase binds. Next, an operator, which acts like a switch. We also have the structural genes, which code for proteins. Lastly, a regulatory gene, which produces a repressor protein. There are two main types of operands. The first one is inducible operand. This operand is normally off, but it turns on when lactose is present. When lactose binds to the repressor becomes inactive, allowing transcription to occur. The second is repressible operand, such as the TRP operand. This operand is normally on, but it turns off when there is enough tryptophan present. Tryptophan binds to the repressor, activating it and stopping transcription. So, overall, gene expression regulation in prokaryotes is fast, efficient, and mainly controlled at the transcription level using the operand system. Next, Nipah will explain how gene expression regulation happens in eukaryotes. Eukaryotic cells are more complex, so their gene expression regulation is also more complex and occurs at many levels. But there is no the factors that cause regulation of gene expression. The first one is because of differentiation, specialization, and development of cells at different times. Other than that, it is also influenced by external and internal environmental conditions, such as hormones, metabolism, and gender. The first way to regulate gene expression in eukaryotic cells are by continuous gene expression. Some genes in eukaryotes are expressed continuously. These are known as housekeeping genes, and they are essential for basic cell function. An example is the actin gene, which is important for cell movement and maintaining the cytoskeleton. The second one is time-specific gene expression. This means certain genes are only expressed at specific stages of development. For example, in fetus, hemoglobin contains 2-alpha and 2-gamma chain. This gamma chain has high affinity towards oxygen for the baby. Later, during infant stages, the gamma chain stops being produced. The last one is by tissue-specific gene expression. Another important type of regulation is tissue-specific gene expression. This means some genes are only expressed in certain tissue. For example, certain genes are only active in muscle cells, nerve cells, or blood cells, depending on their function. In instance, insulin only being expressed in pancreatic cells. This regulation helps maintain cellular balance and allows cells to differentiate, specialize, and develop properly. So, in summary, gene expression regulation in eukaryotes is tightly controlled, involving continuous, time-specific, and tissue-specific gene expression.
Guys, this is our teaching aid named Genie in the Box. Ta-da! Here are the elements inside the box. The first one is the partially loose DNA strand. Next, this is the tight DNA strand. This is the loose DNA strand. There are several substances that will regulate gene expression. So, for the pink one, the pink color is act as the acetylene code, and the yellow one act as a methyl code. For the green color, act as a transcription factor.
So, we move on to the element of like operand. For example, there is promoter, operator, repressor protein, structural gene like C, Y, A, gene regulator, lactose, RNA polymerase. The last one is ribosome.
Firstly, we will give the students to try to arrange the operand system correctly to test their understanding of the operand system positions. If their answer is correct, only then we will continue to the next step.
No!
Next, we will ask the students to show the process of like operand system. Firstly, the presence of the protein, the RNA polymerase can pass through to unwind the DNA double helix. With the presence of lactose, it binds to the repressor protein and causes the protein to differentiate then detach from the operand system. Only then, the RNA polymerase pass through the promoter and operator to unwind the DNA double helix and produce enzymes from leg Z, leg Y, and leg A respectively.
The process of the RP operand is the same with the leg operand. The binding of repressor protein at the operator will block the RNA polymerase to pass through the DNA train to transcribe the TRP gene. With the presence of tryptophan, it will bind and change shape and detach the repressor protein from the operator. This will cause the RNA polymerase to pass through the DNA strand to transcribe the RP gene.
In eukaryotic cell, to regulate gene expression, it involves epigenetic process. When metal group binds to DNA strand, it makes the strand tightly coiled. RNA polymerase can unwind the DNA double helix hence no transcription occur. In other scenarios, when acetyl group bind to DNA strand, it makes the strand loosen tight. RNA polymerase can pass through to unwind the strand and transcription happens.
That's all from us.
If you have any question, feel free to ask. We are happy to help. Bye!